Hello, this is Don Lee, and I am going to share a story that's focused on helping students learn about quantitative traits. So let's call this Learning Quantitative Traits. And again, I am a recently retired faculty member from agronomy and horticulture at the University of Nebraska in Lincoln and teach a 200 level genetics course, or have taught a 200 level genetics course to students that were primarily heading for careers in ag and natural resources. And so quantitative traits are a really important topic. Here's the learning outcomes that we have, and this is the assigned lesson that they can read in our eLibrary. It uses plant breeding as kind of the context for learning about quantitative traits. A lot of our students are more animal oriented, so the story that I will share and emphasize will be an animal focused story, and it'll include both human and it will include the quest to improve an important quantitative trait in dairy cattle. All right, so let's start with this our dairy cattle improvement story. So I have a collection of colleagues from the University of Nebraska who combine their expertise to support the dairy industry, which is, a really important part of the food system. If you love ice cream, you want dairy cattle producers to be successful. Okay, so the question that I pose to the students, and we'll come back to this a little later in the story that I'm sharing here, is this: you have the resources and you have the motivation to buy new cows for your dairy herd, and I'm going to give you information on three different herds, a herd from three different locations that have a priority for dairy: New Hampshire, we'll call them, Wisconsin, and California. Okay, these are kind of hypothetical but realistic, and this is the phenotype distribution we see. And my question is, where would you spend your money to buy cows that will join your herd but also be the future mothers of your herd? Okay, so that's the question, and it's a realistic question given what's going on in Nebraska. Here is Marty, who is originally from Southern California, but he's recently moved and started a dairy operation, a dairy farm near Seward, Nebraska. And a lot of it is because of the urbanization that he and other dairy farmers are experiencing. And there's some advantages to being able to continue in the dairy industry in a different location, a place like Nebraska. And this is a little more recent news that we've got a new dairy processor in this area, and that's really important. If you're going to be a producer in the food systems, you need processors in the food systems, and so the trend has been to have fewer dairy cattle and fewer dairies in Nebraska. The dairies, therefore, are getting bigger and are managed as a bigger agricultural operation. Okay, so again, the focus of understanding the decisions that a dairy producer like Marty is going to have to make means you're going to have to understand quantitative traits. Okay, so I want to emphasize that we bring the topic of quantitative traits and the concepts behind quantitative traits into our students' learning really early in the course. Okay, lesson 2 is when we introduce the concept of quantitative traits, and this is in contrast to what I see in a lot of the commercially available textbooks. So this textbook has organized the book into 5 units, and quantitative and population genetics come at the end. Same with this popular textbook. And when a topic occurs at the end of a textbook, sometimes it's either covered briefly or not covered at all. But quantitative traits are just too important for the kinds of careers that the students that I serve are heading towards. So we want to feature quantitative traits in their learning early on and connect it to the genetics learning that they experience as we start the course. Okay, so both quantitative and population genetics, we don't save them for last. We bring them into the course early on. And so part of the story that we create for the students when it comes to quantitative traits is a quantitative trait that they have firsthand experience with their height, okay? And so actually the very first week we gather with our students in the lab, we ask them to share with us their height data, height in inches, and we put it into a Google Sheets that can be shared and added to, and that's the data that I have available on a quantitative trait that our students are a part of. Okay, so I think that's pretty important, that they're contributing to the learning, being a part of the data set that they're going to work with. And then to lead them into contending with the genetics thinking behind quantitative traits, we introduce this particular genetically controlled trait. Now I'm calling it dwarfism. That's where you can see the mother and the father here. All right, so it's interesting and maybe surprising with these families when the two parents have this achondroplasia condition. They will have both offspring, children that have the condition as well, but have children like their son shown here who would fall into the phenotype category of having a normal height. So this is a question that I'll ask my students as we get to think about how genes work in controlling traits, and the data that I'll share with them will be data that's commonly shared on websites or data from research articles. And so here we can see, if you take a look at the discovery process for the control of this condition, achondroplasia, the gene was mapped in 1994, and then shortly after, the actual gene was identified from that chromosomal region, and mutations in that gene now can be studied in terms of how that single gene can have such a dramatic effect on bone growth in this case in humans. Okay, so if you gather data then from families like this, and you get the height of the height phenotype of the adult children, you'll get numbers that would be something like this. Now these are hypothetical but realistic numbers. So we'll see that there are two phenotype categories. Okay, one of them is about twice as likely to occur as the other. All right, but what's surprising is that this is the achondroplasia, or I'll just use the term dwarf just because it's easier to write. It's twice as likely to be in the dwarf phenotype category than to have that normal height category. We see a 2-to-1 ratio. Okay, so when phenotypes fall into real discrete categories, and when we can see ratios emerge, we want to be able to see if we can apply a genetic model, and you always want to start with a simple genetic model. And so I'll just use a single locus or a single gene pair, and the idea that we have the dwarf and we have the normal condition, and so we see these possible genotypes, and a new idea that we didn't encounter with the experiments that Mendel conducted, a lack of dominance. Okay, if an individual has two of the normal alleles come together, and that forms the zygote, they're homozygous for the normal allele, they'll have the normal height. Okay, if they're from one parent get the achondroplasia allele, what I'm calling big AD with a D superscript, they get that allele plus the normal allele. There's two ways that can occur because there's heterozygous. That will be the condition that we see with the little girl here, the lack of normal bone length in the legs and in the arms, all right? So we see that as twice as likely as having the normal height when we use the simple model of segregation, okay? Well, then what's going on when this happens? When two of the achondroplasia alleles come together, what happens? Well, that's where the lethal genotype comes in. Either prior to birth or shortly after birth, there's a severe developmental effect of being homozygous for the abnormal allele, and those children never continue to live and never reach an adult height. So we see a 2-to-1 ratio in this condition. So there's some new ideas here. Lack of dominance, lethal genotype, but we're building on the ideas that we started with in our entry learning into our genetics course, where we're using the principle of segregation, one pair of genes, two different alleles possible that we see being passed on or segregating in these families. Okay, so we can explain this kind of height variable phenotype with a simple single gene pair or single locus example, all right? So this kind of sets us up then for taking a look at the height phenotype that our students share. Okay, so they gave me this data, and then from this data I can take a collective look at the variability of height phenotype amongst all the students in the class, and I'll get a phenotype distribution that looks like this. So it's different from qualitative trait or a single gene pair type of control of the trait, in that you don't see phenotypes in discrete. Instead, you see them in a continuous. So we see continuous variation, okay? I'll just use the abbreviation there. The other thing is that we tend to see this kind of normal distribution where we see a lot of individuals near the middle and not so many individuals on the extremes, that normal distribution, okay? So when we are measuring phenotypes and we encounter this kind of data, then we aren't dealing with a trait that's controlled by a single gene or maybe just two gene pairs working together. We're looking at a polygenic or quantitative trait. I kind of like polygenic from a teaching point of view. Poly means many, genic means many genes or many gene pairs. So this is what we typically see with a polygenic trait. Now you've noticed that our data actually kind of looks like it has a little peak here and a little peak there. Well, why do you think that would be? Well, our students consist of two gender categories as well as many, many height categories. And if we take a look at the heights contributed by the people in their class that are women versus the people in the class that are men, there is a big measurable difference in average height. The distributions overlap, okay, but there's a big difference in height depending on, did you inherit the X chromosome from your father or did you inherit the Y chromosome from your father? If you inherited the Y, you'll tend to be taller than if you inherited the X, okay? So that there's some genetic component of the Y chromosome that has a big effect on height, but there are lots of other genetic factors that influence variation in height, and those factors influence the variation in the height that we see within and between the genders, okay? So we want our students to understand the genetic basis for why we see this distribution, and the chromosome inheritance alone doesn't tell us the or explain the entire height variation story. So this is a good example of a quantitative or polygenic trait, and here's a good example of a study that was done to try to identify the different loci or the gene locations. Now, my students haven't dealt with the term loci early in the semester, so I'll use the term gene pairs instead. But these geneticists are proposing that there's at least 10 gene pairs, 2 new ones, 2 that have already, some that have already been discovered in prior studies, that have a measurable impact on height variation amongst the humans that they studied. And in this study, they studied a lot of humans, 15,000 or more humans. Okay, so 10 gene pairs associated with height. So that kind of sets us up for thinking about the inheritance of variation when the genetic control of the trait isn't due to just 1 pair of genes or 2 pairs of genes, but in this case, 10 pairs of genes. So then what I'll do is I'll set up a series of questions where the students will build on the learning that they've already done in the course. The principle of segregation, every time we draw a Punnett square, we're dealing with the principle of segregation, and then the principle of independent assortment, where the probability of getting a specific genotype combination at the A gene pair is independent of the probability of getting a certain gene combination or at the B gene pair. And if there's 10 gene pairs here, and I'm looking at a family where we're looking at the segregation of genes potentially at 10 different gene pairs, we can write out the genotypes the way I did. So I want the students to be confident that they can deal with making predictions about multiple gene pairs and use the ideas that we introduced them to and they worked with in the first week of class. So let me just take you through the thinking behind question 21. How many possible different genotypes among the offspring from this cross? Okay, so we're not looking at all humans, but just from this cross where you have, this might be the father, this might be the mother. How many genotypes are there? Okay, so what we need to do is either draw or mentally draw a Punnett square representing every cross. So if we take a look at the A gene pair, the A locus, there are 3 different genotypes here, right? And there are 3 different genotypes at the B gene pair. At the C's, there'd be 3 more. I'll keep track of those here. How about at the D's? Oh yeah, there's both parents are the same genotype. They're homozygous, so there'll just be 1 genotype at the E's and the F's and the D's, and 1 at the G's, but again, 3 at the H's, 3 at the I's, 3 at the T's. And so the overall number of genotypes when you use the principle of independent assortment is 3 times 3 times 3 times 3 times 3 times 3, or 3 to the 6th power. There's 729 different genotypes. Okay, so we're asking the students then to take advantage of Mendel's principles and then just extend this to the idea that the genes at every one of these loci, every one of these gene pairs, is involved in controlling this one trait. We have a polygenic or a quantitative trait. Okay, let's take a look at the next question. Okay, here's where we bring in the idea, it's a new idea, the idea of additive gene action, where the uppercase alleles, in my case, the big A, the big B, the big C, etc., add height, and the lowercase alleles do not add height, okay? And so the tallest possible man or woman among all humans, okay? So we're going to assume that there could be variability at all 10 of these loci, would have how many uppercase alleles, okay? So there's 10 loci, 10 gene pairs, but you could have 2 uppercase alleles possible at each of those, so the answer would be 20. So here's where we get into the concept of additive gene action, but we're applying it to the same data set, we're applying it to the same trait, and so it helps the students add to their confidence because they're building on genetics thinking that they experienced the week before. Okay, so I'll let you on your own work through the rest of these questions, but you can see where we are applying principles of independent assortment and segregation, this new idea of additive gene action, and building on the student's ability to apply these to an important trait, in this case, variability in height, when it's controlled by lots of genes having an additive effect. Okay, I should mention that I tend to use a simpler explanation for additive gene action where every uppercase allele adds the same amount of height. Okay, it's not always the case, but in learning the concept, that tends to be the better case. All right, so with those questions so far, we can see, because there's lots of different genotypes that control this trait, and they're using additive gene action, we can understand why we see so many different phenotypes. So one aspect of the quantitative or polygenic trait, lots of different phenotypes can be understood by applying those concepts. The other concepts we want to understand is that why there are so few individuals in the extremes and there's so many near the average. Okay, that's another one. And so here we get at the average height. Okay, the average height of a man or a woman will have not the maximum 20 uppercase alleles, but 10 uppercase alleles. And there are many different ways in which you can have 10 uppercase alleles. You could have, all uppercase alleles at the A's, the B's, let's see, the C's, the D's. What do I have so far? 8, and there's 10. And then the lowercase alleles for all of the other 5 loci or 5 gene pairs. But that's just one of the many ways in which you could end up with 10 uppercase alleles. So there's a lot of different combinations of genotypes that would give you an average or near an average phenotype. But how about the tallest or the shortest? If the tallest has all uppercase alleles, then that would only be occurring a quarter of the time at the A locus, a quarter of the time at the B locus or B gene pair, etc. And so there would be a very low probability of getting that extreme. Okay, so just thinking again about the principles of independent assortment occurring at lots of gene pairs, and adding in the concept of additive gene action, we can understand why average is quite common, extremes are quite uncommon when you're dealing with a quantitative trait. Okay, so that's what I'm getting at with these next two questions. All right, so you can see what I'm doing there, using a quantitative trait that they're familiar with, their height data set that would represent data that they would be contributing to, data that they helped collect, and we're applying concepts that they've already experienced as we learn about quantitative traits. All right. The other aspect of quantitative traits that we want to understand is making predictions with quantitative traits. Okay, and so because of the importance of a quantitative trait like milk production, geneticists are very interested in doing what cattle geneticists are very interested in doing what the human geneticists did, and that's discovering where those genes are located that are variable and having an influence on this important quantitative trait. So then that leads us back to the story I told you I challenge our students with in this class time when we gather together in the lecture hall. Again, so you are a dairy operation manager. you've got cash because you've sold land in Southern California, and you're moving some of your cattle, okay, to Nebraska, and you need to buy more cattle, and those cattle will join your herd and be the mothers of the future members of your herd. And if you've got access to milk production records from 3 different herds, we're saying one herd's from New Hampshire, one herd's from Wisconsin, one herd's from California, where would you invest your money? If you know which cattle are producing the most, all right, and because you have access to the records, are you going to buy the best cattle from New Hampshire, the best from Wisconsin, or the best from California? And what's interesting to me when I pose this question to my students, I get a variety of answers and a variety of justifications, okay? And so to me, it seems like the obvious answer would be that, well, the New Hampshire breed has all of these cows that are contributing to this high milk production. Those are the cows I want to buy. They have the highest milk production phenotype. But I do get students saying, well, what I like about the California population is that it's consistent. Okay, so one of the questions that I'll ask the students at this time is, how would you describe the distribution in these two populations? What are some of the statistical measurements we can have? Well, one measurement they've learned about is the average or the mean. So which one of these populations has the highest mean? Well, that's a trick question. They all have about the same mean. They all have the same average, but they don't look the same. What's different about their distributions? They have a different dispersion around the mean. They have a difference in their variance, okay? And so some of my students have had statistics, some of them haven't, and so some of them have calculated variance, some of them haven't. I just want them to recognize what each is measuring. Mean is measuring the central tendency. Variance is the distribution around that mean, and obviously the New Hampshire has a bigger variance. Okay, so if we're interested in genetic improvement, then you want to have variation. And so it seems like the New Hampshire breed or individuals would be the best choice. But there's more thinking that one needs to do to be confident in your decision. So what I like to do is kind of turn the distributions into something that's a little more visual. Okay, so each of those values that make up the curves is the production level a particular cow is producing. So here I'm showing the cows from the 2 different herds. Same number of cows, just a different difference in distribution, okay? And so again, you're the dairy professional. You're deciding, where am I going to go and buy a cattle that'll bring in the phenotype, the ability to produce milk, but also bring in the genes that I hope are desirable versions of the genes that control milk production, and so my herd can get even better. All right? What are the questions the dairy professional is going to have? Okay, so they're going to know that the variability in milk production phenotype is really due to 2 different things. One is because this is a genetically controlled trait, it'll be the genotype of the individual. How much of the variability is due to the genotype, okay? But there's another factor that would have an influence on the variability, the phenotypic variability, okay? So if I've got the money to purchase some of these cows, here the cows that I would purchase from the California herd aren't a whole lot different from the average, okay? Here, these that I purchased are quite a bit better than the average, okay? But what is that difference due to? What does that variation due to? Is it due to the genetic difference from the cows, or is it due to something else? Well, what else could it be due to? It could be due to the environmental differences. Environmental differences, you could think of them as those factors that the dairy manager, the dairy professional, can control. How healthy do you keep the animals? How do you monitor their feeding? What kind of a climate do they live in throughout the day so that you don't put stress on that? All of those factors that a dairy herd manager can control all roll into the environmental category, and variability in how those animals are treated could result in variability in their milk production. Okay, so the question this manager would have is how much phenotype variability, or how much environmental variability was happening in the California herd management compared to the New Hampshire herd. What if these high-producing cows were the favorites of the dairy herd manager? He or she made sure that if they looked like they were a little sick, they were tended to with their medical needs. They always got the best food at the optimal times. They were just cared for using more of the resources and time, and therefore those that were on the low end suffered. They didn't get the same level of care. So was there more environmental variation or in the New Hampshire herd, and does that have an effect on the phenotypic distribution? So what we're really interested in is a concept called heritability, and in our case it would be the broad sense heritability. And here's the formula for broad sense heritability: genetic variance over phenotypic variance, okay? And so we can see that the New Hampshire herd has the most phenotypic variance. The California herd has the least phenotypic variance. And, our basic biology tells us this: the phenotypic variance, the phenotype, is because of the genetic variance and combined with the environmental variance, all right? And so if we can measure the phenotypic variance as we measure milk production, how do we get a read on how much of that variability is from the genetics and how much is from the environment, okay? If we can do that, we can calculate heritabilities, and we can use heritabilities as we go about making selections or making predictions, okay? So the key here, the key idea that I like to share with the students in our course is one way to calculate broad sense heritability is if you can measure environmental variance that is occurring between genetically identical individuals, okay? And this is what is done in human studies, and it can be done in other studies. It's very common to do in plant genetic studies because we can generate lots of plants that have the same genotype. In animals, you're kind of relying on the occurrence of monozygotic twins. They have the very same genes. They may not have the same phenotype. If there is a phenotype that's different, it's due to the environmental impact on how their bodies grew, developed, and are expressing their traits. So if you can measure differences between identical twins compared to individuals that aren't related, you can get a measurement of the environmental variance. Okay, so we're going to make the assumption that we have in our herds monozygotic twins, cattle that are genetically identical, okay, living in our herd producing milk, okay? So there's the two genetically identical twins in our California herd. There they are in our New Hampshire herd. And what do you notice about their phenotypes? They're not very different. There is some difference between them, though. What would that difference be due to? That difference would be due to the specific environmental factors that had an influence on milk production. They're not living in exactly the same environment, therefore, they're experiencing the same environment. Therefore, there's some phenotypic variance, but it's not due to genetics. They have the same genes, the same versions of those genes. It's due to environment. So if we can get a measurement of the differences between lots of different twins in a group, we can get a measure of the environmental variance. Okay, so again, we'll just apply it to our simple idea here. We've got more phenotypic variance in this New Hampshire herd, so I'm going to give that a, and I'm not worrying right now about calculating environmental or these variances. I'm just giving them these values. Okay, so we have 8 here. What should this number be, this phenotypic variance? Well, it should be less than 8 because it's a smaller amount of milk production variability, so I'm going to assign it a 4. We have in both situations about the same amount of environmental variance occurring. Okay, our identical twins have the same difference in their phenotype, so I'm going to assign 2 to the environmental variance. So what does that leave us with? Our genetic variance here would be whatever that number is plus 2 gives us 4. Here would be whatever that number is plus 2 gives us 8. So we can just do the simple math there. There's our genetic variance. And then we can apply those numbers to our broad sense heritability. Genetic variance over phenotypic variance, which is genetic plus environmental variance. So here it's 2 over 4, or a heritability of 0.5. Here it is 6 over 8, or a heritability of 0.75. Okay, so what does that mean? If we have this heritability, this higher heritability, and we're selecting these individuals here, we are more confident because of that high heritability that they're different from the rest of the herd because of the genes that they possess. There's a small amount of the variability that's due to environmental differences. Therefore, selecting these, we should be quite confident, and knowing that the heritability is high, we should be quite confident that they have the genes we desire for higher milk production, and they will pass combinations of those genes on to their offspring. This would be a better choice. These individuals that we select with this lower heritability, they may have some more desirable gene versions for higher milk production, but they're not much different genetically than the rest of the group. A higher percentage of the variability we see in this group is due to environmental differences. Okay, so heritability doesn't give you a probability statement. It does give you an idea of how successful do I anticipate being if I want to make improvements in this trait. In this case, higher heritability and selecting those individuals with the desired phenotype, you'd be reasonably confident that you have a chance of maintaining and improving that phenotype in future herds. Okay, let's change our situation here a little bit. We won't change our California herd. We'll just change our New Hampshire herd. What if our identical twins are these 2 individuals? What does that mean? Well, we don't have any difference in our phenotypic variability. It's the same group of individuals, the same phenotypes that we've measured in milk production, but those identical twins are much more, have a bigger difference. And that means that the environmental variance is much higher. I'm going to give that a 7 in our hypothetical example. What does that mean? Then our genetic variance is quite as making a small contribution to our phenotypic variance. That would be a 1. So now our heritability is 1 over 8, or 0.025. So even if I would select the very best producers, I'm not as confident that they have that milk production phenotype because of the genes that they possess and express. It might be that they have that phenotype because of the environment they experienced. I'm not as confident that I would make progress with in selecting these high milk producing individuals. So that's the kind of decision making we have with quantitative traits. Is we don't assign the same kind of probabilities that we do with qualitative traits, but we can make these generalized predictions about the opportunities to make improvements. All right, before I move on, then I'll leave you with this just one more look at this image. You could kind of adopt this approach, this teaching approach, with lots of different traits and lots of different living things. Again, I think if the students are just getting started in working with statistical analysis and are starting to get a handle on what these statistical measures provide in terms of biological insight, making the example more visual this way can be helpful to those kinds of students. Okay, I wanted to mention that to give our students experience in calculating variances, we take advantage of the tools in Excel, and so if you've got a file that you can create into an Excel file, you can actually calculate your phenotypic differences here using some of the tools that Excel provides. Okay, doesn't really help the student, I think, understand what variance does compared to just calculating it by hand. But we definitely want to build within our students' confidence in working with data and using tools like Excel to do that. And this gives us an opportunity to show them you can calculate variance from our data. That's phenotypic variance. And if we take advantage of prior studies to get an estimate of environmental variance for a trait like height, we could make assumptions that that's at play in our population and calculate broad sense heritabilities. And that's something that we ask them to do then in problem sets and in take-home exams. So they get a little bit of experience working with the data that we collect, the way a quantitative geneticist would work with data. And that's really our goals, is to have an opportunity to learn genetics by working like a geneticist. Okay, so with quantitative traits, very important our students engage in thinking about quantitative traits early in the course. And so comparing them to qualitative traits, understanding why we see that normal distribution, the concept of additive gene action, and the concept of broad sense heritability are our learning priorities. All right.